Stem cells are really pretty interesting. And anyway, it's the last, 5-6 years has seen an enormous increase in the role of stem cells in cancer growth. Not only has there been a great increase in the growth of research in stem cells, there's been a lot of metastasis, because it's used to start in Britain and the United States, but it's now a lot of activity in China and elsewhere. I never really understood cancer terribly well, and I'm still not sure I understand it completely, but I think if you look at cancer from the point of view of stem cells, it makes a very big difference in terms of understanding the processes that are occurring. So what is it if you look at, oral cancers, head and neck cancers, the same as other cancers in many ways, they've got their own peculiar features. But if you look at, shall we say, oral cancers, what sort of things is it that occur and what are you trying to explain? Well, first of all, if you take pre-malignant lesions and early cancers, they're clonal. Many of them, if you do PCR, you can find that all of the cells within the lesion have originated from a single cell in a single series of continuous mutations. So one cell is spread to occupy this entire territory, which is a little difficult to understand. Secondly, the lesions show progression. This is one of Eric Dallaston's patients who came in with a rift plaque, was told to come back again, and didn't come back for 2 years, by which time it was turned really nasty. So there's progression of these lesions as well as spread. And then, of course, perhaps the two most serious things from the point of view of survival is that following therapy, cancers often shrink, but then they come back. And of course, why do we do that? Why do we have recurrent sperm therapies, which we've been working on for 30, 40 years, are supposed to be effective? And then, of course, of particular interest is lesions metastasize, head and neck lesions to victims and lymph nodes. But this, of course, is likely to be a major problem in terms of survival. So what I'm going to suggest to you today is that stem cell phenomena are potentially associated with each of these. I'm going to talk about this a little, and about this, and about this. Okay, so I expect everybody knows about stem cells because they're in the newspapers all the time, but I thought I would just refresh your mind about what stem cells are and why they're interesting. Everybody here was an ovum, I guess, Ian, because the ovum formed at birth in their mother. But anyway, if you get a fertilized ovum here, you get a bunch of cells, and they're totally potent. They can do anything, okay? You can split them up, you can get 4 embryos out of this one, but as they grow, they become differentiated, they form blastocysts. The blastocyst consists of 2 parts, really, the inner cell mass and this outer cell layer. This is the bit that's going to form the embryo, and this is the bit that if you take it out of the blastocyst and put it in a dish, gives you embryonic stem cells. An embryonic stem cell can grow pretty much indefinitely, and you can turn them into pretty much any tissue you want. Not as easily as it may be made out sometimes, but generally so. Later, you can still get such cells from other parts of the fetus, but when you're grown up, even if you're as old as Ian and me, we still have stem cells because we're still making new tissue. We're still wounding, repair, and so on and so forth. But we get 2 lots of stem cells here. Tissue-specific stem cells, one for liver, one for eye, one for skin, one for bone, and so on and so forth. And then we also have a few multipotent stem cells. We've got some of the residual property things lurking in various places like dental pulp and wherever else the Lucy may tell you about. But anyway, lots of stuff like that going on. But what we've been very slow to realize is if stem cells are the basis of generation to generation of all tissues, which they are in the normal body, then they're also presumably the basis of all malignant tissues. So we have another category up here, which is malignant stem cells. Okay. So what is it that's so magic about a stem cell? The answer is there's nothing really magic about a stem cell. It's just got 2 interesting properties that characterize stem cells, which ever saw, except interestingly this one, never mind. Stem cells are characterized by indefinite self-renewal, so they can divide pretty much forever. They can divide longer than we live, typically. And as they do it, they have the ability of shedding other cells away from the stem cell compartment, which go on to differentiate to produce cells for tissue function. And that really is all a stem cell is, whether it's in an embryo or in later things, or, for that matter, in a cancer. So why is there a bit of a problem in getting stem cells in cancer accepted as a phenomenon? Well, basically, this is a piece of epithelium, placal epithelium, it's human, and these are the rete. And if you look at the rete, stem cells are localized in the tip of the rete here. This is a marker of stem cell zones. These are not all stem cells, it just marks the zone. This is differentiation. All of the epithelium is differentiated, except this little bit here. So we can be pretty sure the stem cells are in this zone. That's where this is. But of course, when you get malignancy, you lose your anatomy, you get a real mess. And I think people assume that your stem cell pattern was lost because your anatomy was lost, and because the stem cells normally live in these little niches. But it's apparent that if you follow my definition that I made a few minutes ago, that a stem cell is a cell that can renew itself and produce differentiating cells, you know there are going to be stem cells here because this proliferates forever, and what's more, nearly all tumours, except the most anaplastic, are going to be producing differentiation features, easy pearls, and bits and pieces. So we know there are stem cells there. So the interesting thing is not whether there are stem cells, but whether the stem cell pattern, the hierarchical stem cell pattern, which is present in normal tissue, is present in cancer. And this pattern looks a bit like this. You have a stem cell. This is in a homeostatic, a steady state, when the tissue is just renewing itself in normal health. The stem cell renews itself, it divides to produce two cells. One replaces the stem cell, the other one differentiates, proliferates some more to produce a lot of differentiating cells, okay, for tissue function. So the stem cell divides rarely, it produces a lot of cells, and it's different. It's got different cell cycle controls, it's got different survival mechanisms, and various other bits and pieces. So basically, how could you tell whether this sort of pattern persisted in this sort of lesion? Well, the experimental way of doing that is to get your cells and take them out of the lesion, mix them all up, and then stain them for markers which are potential in either positive markers for stem cells or by staining amplifying cells and negative markers for stem cells. And if you can do that and put them back in a mouse, you can have prospective identification of tumor-initiating cells which correspond to stem cells. And the way you do that is just diagrammatically represented here. You get your cells, you stain for a marker, you put them in the port of the mouse, and if this one gets a tumor and this one doesn't, you know that these cells have got cancer potential, and these happen. These are stem cells or tumor-initiating. These are non-stem cells, non-tumor-initiating. So the gold standard for identification has been injection into mice. We don't really believe that that is the only way of doing it. And our work has been based largely on how stem cells divide and how stem cells remain stem cells once you put them in a dish in culture. Now you remember the picture I showed a few minutes ago, showed the early embryo, okay? Where you had the stem cell, the ovum producing stem cells and producing a bunch of stem cells, all right? A very good thing when you're developing and you get a lot of stem cell expansion, okay? But that's not much use in getting a person because you've now got a bunch of stem cells, you haven't got bone, tissue, eye, whatever specialized tissues required. So the usual way was the way that I showed you on the previous slide, where a stem cell divides to produce two cells, one which stays a stem cell and one which differentiates. And this is called asymmetric division. But basically, if a cell divides like that, you get a steady state. The number of stem cells remains constant, the number of cells forming new tissue remains constant, and you've got a relatively healthy tissue. If that guy switches back to this, you've got a problem because you're producing more stem cells, which are producing bigger lesions. The other thing, of course, is that your differentiated cell here, or your stem cell for that matter, will produce two differentiating cells, whereupon it dies and it's gone. But you can see that technically, if you did make this cell produce two differentiating cells, you wouldn't have a stem cell. So if this was a cell causing the cancer is going to disappear too. Okay, so what do these things look like in the dish? Well, they look very pretty, actually. And these are cancer cell lines that we've got from cancers that Ian has given us. And basically, if you plate the cells out very sparsely, so one cell, each cell can be seen what it forms as clonal growth. Some cells form very tight little rosettes and continue to do that as they grow. Others form cells that are bigger and a bit looser and form raggedy colonies, and then some hardly grow at all and they're bigger and sort of cells. These are the stem cells, and if you look at them, they're characterized by cobblestone growth and expansive growth, and they're called holotones. Okay, then these are the amplifying cells here, and these are called mericons, and these are the late amplifying cells, and these are called paracons. So we can actually identify the stem cells in the dish when we look at them. And we showed, about 5-6 years ago, that this sort of pattern is present in all sorts of tumors, breast, prostate, head and neck, and bunch of others. What does it do? So you take your stem cell fraction, and you can take one cell at a time out of there, and put it in a dish, and it'll form a new cancer, new cell line, and this can be grown forever. You take these guys, they don't really form anything, they just die off. So we now know which the culprit is. This is the guy that's causing the tumor to grow. This is the cell that has to be spread if you're going to get metastasis. Can you identify it apart from just seeing it? Well, yes, you can, because it's a stain something called CD44, which is a hyaluronate receptor, which seems to be expressed in high levels on most epithelial stem cells. So if you stain two colonies like this, here's the paracline, here's the oricline. You see the dense nuclei, big loose nuclei. This one here stains for CD44, so this is, if you like, a stem cell marker. It marks the population that contains the stem cells. These are probably not all stem cells, but a large fraction of them will be. So now that you can do this, you can use a machine called fluorescence activated cell sorter, okay, or flow cytometry, it's also known. And what you can do is you can set the machine up to find the green ones from the non-green ones. These are the green ones, about 3% of the cells in this tumour, and here's another bunch over here. And so basically, if you take these cells, you can show that they form these spheres, so they can survive in suspension. They can grow when other cells can't, which is an interesting from metastasis. And our assumption has been, and it's now shown by lots of other models, that these cells, the CD40 high cells, are enriched for cancer stem cells. And that's the population that you've got to kill if you're going to keep on solving cancers. So how do we explain this? That this whole lesion, okay, is formed probably from a single stem cell. Each stem cell forms a little territory like this. This is human mucosa. We've taken it out, we've transduced the stem cell with a marker, alpha phosphatase. You can see that each stem cell makes a little territory. So how does that little territory, which is about 40 microns across, turn into this bloody great lesion? Well, the answer is that the stem cells have to multiply in some way, okay? So how do they do that? Well, we're not going into this in great detail. There's a guy called Doug Brash in the States who's looked at this for skin cancers where it's much easier to look at them, and this is done in mouse. And what he shows here is that these little units, each one of these units, when it gets pre-malignant, it expresses a form of P53, which is a cancer-related thing, which is stable. So you can stain for it. And if you continue putting UV on these ears, then they grow big. So what you're doing is, it's not one unit, it goes 1 unit, 2 units, 3 units, 4, and they grow as units, okay? And so the concept is that what's happening is that this is skin and mouth, each one of these is a unit, and each one of these has a cluster of cells related to it, and in the middle of each of these is a stem cell, okay? So let's have a mutation occurs in the stem cell, say this one. What's going to happen? Well, that cell is now going to produce the other cells, so all of the cells in that unit are now going to have a mutation. And it turns out that that's very stable, because there's good defenses of these units around the edges. But what happens if we kill one of the other stem cells? Okay? If you kill a stem cell's got to come back from somewhere else, and it comes back either from here or from here. But the chances are that it comes back mainly from the cancer stem cells, as I'll tell you in a minute. So it spreads, and it can spread again and again and again. And these stem cells take over as a sort of territory. So the problem we're now facing is that if this is to occur, you have to postulate that these guys are in some ways tougher than the other cells that they're replacing. So because these are being killed for some reason, and this probably works in the mouth, where you get your initial mutation through tobacco smoke or something of the sort, and then you're getting promotion of the tumour through alcohol, wounding, damage, various other bits and pieces that can occur. So that can explain the sort of spread that we see. The other thing, of course, is that you probably all know that cancer is a disease of multiple genetic hits. Okay? So the chance of a cell getting a particular genetic hit may be very low, 1 in 100,000 or something, right? So a chance of getting a second hit, which is the right hit, is very low indeed, and gets lower and lower. But if you've now turned your one cell into 100,000 cells, the chances of one of those cells getting the second hit is very high. And so by this stem cell field promotion, you get a really much greater chance of cancer. So how can we demonstrate that? Well, here's your holoclone, and here's a paraclone, and we've irradiated it. And if you look what happens to the holoclone, not much happens. Those cells are resistant to the radiation. What happens to the paraclone? Half the cells are killed. So it looks as though these cells, the paraclone, non-stem cells, are much more susceptible to therapy than the other guys. How do they die? Well, you can stain for this thing called a nexin, which is a marker for apoptosis, and they're in apoptosis and dying through what's called programmed cell death, which is the way that most of the cancer therapies, either chemo or radio, kill cells. We can actually do numerical analyses of these using that same machine again, the FACS, okay? And what we do is we stain these cells for 3 markers. We stain them for a marker of an nexin, which is a marker of apoptosis. We stain them with DAPI, which is a marker of cell death. and we stain them with CD44, which is a marker of being a stem cell or not being a stem cell. And if you do that, Dappy is dead cells, so everything above the line is dead. The next thing is apoptotic cells, so everything to the right of the line is apoptotic. You can divide your population into 4. Happy, nice cells, early apoptotic cells, late apoptotic cells, the cells will be dead for quite a long time. But now, if we take that population and stain it for CD44, which is our stem cell marker, we take the top 5% here, okay, and we look at whether these cells are dying or not after therapy, we find that instead of 20 plus cells dying percent, it's less than 3%. So they're more than, the stem cells are more than 10 times more resistant to standard therapy procedures. So the chances of killing these cells is not terribly good. So you can show that with a whole bunch of therapies. So everybody who talks about cancer stem cells is a dandelion picture, okay? And this is really pretty, but it's not really science, but I like it, so I put it in. So basically, when you mow your lawn and you come along and you chop down your dandelion, you are not doing very much good, because the regenerative capacity of the dandelion lies in the root, the stem, in that region. Okay? So what happens is it comes back again. So what happens if you treat this stem cell hierarchy that I told you about? Okay? Here's your cancer stem cell, early progenitor cell, and then your differentiating cells. Well, from the data I showed you a few minutes ago, you know that these cells are susceptible and these aren't. So you treat the tumour, and the tumour gets much smaller and virtually vanishes, because these are probably less than 5% total population. But of course, that's where all the regenerative capacity is, so it's coming back again. A bit of a problem, I think. What can we do? There are various things which I haven't got time to go into. But basically, I think one of the interesting approaches is to get this guy to form 2 differentiating cells. So you wipe them up, but you don't kill it, you differentiate it. And that looks as though it's a strategy that's going to work. So this is a very, Chinese friends tell us that one showing is worth 1,000 tellings. And what we have here is a holoclone, which is a stem cell colony. And here we have some other cells, which are not stem cells. I'll explain a bit later why they're moving around, but not yet. But if you look at this, I hope the video works, what you're going to see is the cells are dividing here rapidly. But the stem cells are stationary. They're not going anywhere, okay? They just stay there and divide locally. Whereas the other cells around here are having fun. They're sort of going round about. Here they are. Call it this guy. They've undergone something called EMT, epithelial mesenchymal transition. They've stopped being epithelial, they've become mesenchymal, but they're still cancer cells. They're still stem cells, okay? And you can probably see that they're actually being formed around the edge of this colony, around the edge, like at the edge of a tumour. You've got these little cells which have undergone this change and are beginning to drift out, and these are the ones that are going to metastasize. You can see these here. But these cells that have left are still dividing. Some of these will divide. There's one that's dividing. One of the motile ones is dividing. They do divide. They just don't divide as fast. Anyway, how do I get out of here? There. Okay. A guy called Bradlett's, a very good guy, came up with the idea that what's actually happening here is that for cancer to metastasize and spread distantly onto the local lymph node, it's got to stop being an epithelial cell, because epithelial cells are stuck together, that's how they function, and turn into a motile cell. And he's got this idea that there are motile stem cells, okay? And so basically we started looking at that, and it's quite apparent that if you look at early cultures of stem cells, okay, there's a heterogeneous bunch of cells in there, and some of them look EMT, they look like fibroblasts. If you stain a colony like this for a marker called vimentin, which is a classical marker of EMT, you can see these cells on the left here are producing these cells, and these are moving out in front. So we think that is what's happening in metastasis. So I've shown you the real-time video, and it suggests that individual cell morphologies, you can recognize these guys, and the types of colonies they form, the motile things. So we can now actually look at the dish and then hit them with various things, medicaments or radiation doses, to see whether we can selectively kill one or the other. We can selectively kill these 2 guys as well. The problem is that we now have 2 lots of stem cells. 3 lots of stem cells. We've got a stationary stem cell, which is doing most of the growing, which probably corresponds to the bulk of the tumour. And then we've got this EMT population, which is probably what's zooming out into the tissue. They're both stem cells. These can revert back to that. Those can make those, so they can switch around and avoid whatever you're doing. So you've got to find some therapy that kills both of them, or you're going to be in trouble. Can you identify them? Yes, you can. Same old machine, facts. We get three populations. This is CD44 and another marker called ESA. This is the motile stem cell fraction here, and this is a fresh tumour. You can see something a little bit similar. It's much more difficult to do fresh tumours. Helena, who's a PhD student here, is trying to do it. With Ian's help, we get tissues, and we're trying to work out how the fresh tissue functions as well as cell lines, because that's important. But basically, if you take these, they form these. If you take these, they form those. So we can now focus in on the two stem cell fractions. Interestingly, they respond differently to therapy. I haven't got time to go into the slide, but Adrian Bill, close up to the lab, have done that. And you've got different colours here, and you've got paclitaxel and cisplatin. These are cells which have got various, they're clones of cells that have got various stages of EMT. Okay? But you can see that they respond with the pink line, which is paclitaxel not being killed, cisplatin being killed, this one not being killed, not being killed here, being killed here. So the whole thing is getting very complicated, and we've got to get a bunch of assays together to work out what's going on. So what we think is happening in a tumour now, which explains, I think, a lot of my problems that I've had in understanding what's going on, is that you've got a bunch of cells within the tumour. Most of them are this holoclone stem cell fraction. They produce cells which go down a differentiation pathway. That's the normal pathway, but because they're in culture or because they're malignant, they're not doing it very well. But that's the pathway that we thought was important once upon a time, and we still do, because if you get this cell to do that, you get rid of the generative capacity of the tumour. But what they found out is that these cells also form these motile cells, and these motile cells can float, and if they do float, they can form spheres of tumour cells, so you can understand that that's a nice property to have if you want to zoom around in the lymph or bloodstream. And this whole lot is interchangeable, going backwards and forwards and backwards and forwards. So the problem now is to work out the individual functions of these cells, what they're doing in vivo, see if we can get our in vitro work, compare with our vivo work, and see where we go from there. So basically, in summary, I think it's absolutely established now that stem cell patterns exist in tumours and that stem cell patterns persist in the laboratory in the dish. It seems from what I hope you understand and believe me that tumour recurrence is associated with stem cell resistance to therapy. Okay? And so, as I said every time to Ian, it's great to be a surgeon because with a knife you kill everything. Okay? The one therapy that is not selective is surgery, because you just take the whole damn lot out of the fucking form and it works for me. Okay. Spread and invasion of tumours. Invasive tumour trump, which everybody's been interested in for years, appears to be related to EMT-like changes at the stem cell level. Okay, the stem cells are going in and out of EMT and enabling themselves to wiggle around and get out of there. And that it's the stem cell population in each of its phenotypic manifestations, stationary, motile, maybe there's another one we don't know about. And it's each of those manifestations which need to be eliminated if you're to eliminate the tumour. Okay. Research questions, since I know you guys are all just dying to rush into the lab. We don't really know to what extent the behaviour in the dish reflects the behaviour in the living tumour. Okay? It's very difficult to work with living tumours. We're very fortunate in having Ian to give us tissues that we can look at, and we're beginning to work on that. But it appears that it does reflect pretty much the basic stem cell patterns and the drug response do tend to be there. Of course, having mesenchyme around the tumour influences it by projection of cytokines and other bits and pieces. And we know that cytokines increase that EMT, hypoxia increases that EMT. The great thing about culture is you can actually control the hypoxia. It's not just hypoxia around the edge because the vessels aren't full. What induces EMT and leads to invasion? Well, as I said, inflammation will do it, damage will do it. If we're interested to know how much damage done by surgery may induce cytokines which induce an EMT change, which suggests spread. We don't know that yet. Hope it's not much. What's the role of hypoxia? Because hypoxia, tumours are usually hypoxic. It looks as though hypoxia is a great stimulus to tumour invasion because it induces HIF1 alpha, which induces EMT, as we've got a student in the lab room working on that, and that goes really well. Interesting thing, which nobody knows, is not in the literature at all, and if we've spent some time looking for it, we've tried to examine it, is that if this thing goes into a motile form and then whips off to the lymph node, what is it that turns it back into the epithelial form that allows it to make a secondary tumour there? because you know that the secondary tumours histologically look like primary tumours. So there's this reversal going on. It would be really interesting to know what mesenchymal epithelial transition, how that occurs, because I think that's probably a big rate limiting step on the formation of secondary lesions away from the primary tumour. Why are some tumours controlled by chemoradiotherapy? Yeah, if everything I told you was true, we'd never cure a tumour except by surgery. And yet it does appear that some tumours respond. So it appears that we've probably not got it completely right. There are combinations of drugs or particular drugs that will affect stem cells. And there's been some work done in the States which suggests that you can identify things which selectively will kill the stem cell population. Unfortunately, with the exception of etoposide, which is used as an adjuvant, the reagents used are not clinically effective. How are combined therapies to be developed and tested? And that really is a prime thing for opportunity for research between clinicians and the laboratory, because this really is a clinical interface. And finally, I think what every other tumour is really moving to, with the data I showed you, shows that cell lines differ in their responses to drugs. It may be that primary tumours do too, and it looks as though we're going to need to design particular drug schedules for particular tumours. So we've got to get away from getting a biopsy out of a tumour, scanning whatever genes or whatever pattern of stem cells is going on in there in order to correctly diagnose the appropriate therapy for the individual patient. So that's all I'm going to tell you, except these are the guys who do the work. I've got 3 visitors. Helena's not really a visitor, she actually works here, but we've visited from, we've got Helena from Brazil and Hideo from Japan. We're working with us on these clinical problems. I've got a bunch of books in the lab with students. We've got Kim Piper and Ian Hutchinson, who've done sterling work providing specimens. We've got funding, fortunately, from Saving Faces and NC3Rs. And we've had funding from DBSRC, breast cancer, in fact, anemia. And I thank you for your